Welcome back to Catalyst University. My name is Kevin Tokoph, and in this video we're going to talk about a really cool supplement called L-Theanine. And L-Theanine, just to put it simply, is a non-sedative relaxant. Alright, so this is the molecule theanine right here. And theanine is an amino acid. It is a plant-derived amino acid, and humans do not make this naturally. We do not make this. In fact, this amino acid is not in proteins in any organism for that matter, and for humans it's not required for life. You could get by your entire life without this molecule as far as science goes, and you'll live a perfectly long, healthy life potentially if you follow all the health rules and so on and so forth, etc. But it turns out that theanine can be taken as a supplement, and it has a lot of cool effects. Now, a lot of times when you take something to relax, what do they say? They say take the chill pill. So in previous videos, we've talked about benzodiazepines, we've talked about ethanol, barbiturates. Those are all GABA receptor agonists, meaning they promote hyperpolarization of membranes, and they do act as a relaxant, but really what they're acting as is a sedative. And so we have this conflict in terms of taking things, whether it's drinking a beer, taking a benzo. How do we achieve relaxation without being sedated? Because for the most part, those other things I mentioned, barbiturates, benzos, and alcohol, put you to sleep. Well, it turns out there's a supplement, theanine, and it does not act as a sedative. In fact, if you ever take theanine, you're never going to just get zonked taking it, but it does relax you. And we're going to talk about why that is. Now, the other ones I mentioned, barbiturates, ethanol, so on and so forth, those are GABA receptor agonists. We have this concept in physiology that in the central nervous system there are two competing neurotransmitters mainly. There's glutamate and GABA. Glutamate is the excitatory neurotransmitter. It binds to its receptor and it promotes excitation. GABA is the inhibitory central nervous system neurotransmitter. It binds to its receptor and it promotes inactivation or relaxation of neurons in the brain, etc. Okay? So it's a balance between the two. Now the reason that those three molecules, benzos, barbiturates, and ethanol, promote sedation is because they're directly agonizing or acting as agonists on the GABA receptor. But the way theanine works is very different. It does not do anything with the GABA receptor. It's an antagonist of a glutamate receptor known as the NMDA receptor. So it's an NMDA receptor antagonist. So it does that and it blocks the action of glutamate, but also through a different mechanism we'll look at in a couple of slides, it actually decreases the available glutamate in glutamatergic neurons in the brain. And this is just one of the supplements that you can buy for theanine. They promote stress management, and we'll talk about that. And actually, if you look at a lot of sources, there's a lot of evidence, robust evidence, from multiple studies that have indicated that it is efficacious over direct supplementation of GABA. And GABA also, you can buy GABA in a supplement like this, but it doesn't work. And we'll talk about why that is in a couple of slides. All right, but for now, let's actually talk about how theanine is synthesized in plants, because this is a plant amino acid, one that they make for their own purposes. And in fact, theanine is made in particularly plants of certain tea leaves. People always drink this green tea to relax. Well, this is why. All right. So it turns out that theanine is made from two molecules, glutamate and ethylamine. Now, glutamate obviously is a normal everyday amino acid floating around cells, but the ethylamine, how do we make that? Well, it turns out you need alanine to do that. And there's an enzyme, pyridoxal phosphate dependent, called alanine decarboxylase, which removes the carboxyl group from alanine and just leaves ethylamine. And then through the action of this enzyme, theanine synthetase, which is an ATP dependent enzyme, it condenses ethylamine and glutamate to make theanine. And again, it's the L isomer of theanine. That's why I indicate L-theanine. All right. Theanine synthetase is similar in function to glutamine synthetase. The only difference between the reaction mechanism is instead of putting ammonia in to make glutamine, it puts ethylamine in, a similar molecule. All right. So this requires two amino acids, alanine and glutamate, but it also requires an ATP. All right. Now there's another way that plants can do this, and it's actually through the enzyme glutaminase. It turns out that glutamine, we can start with glutamine, and instead of hydrolyzing off the ammonia right here, instead of using water as the other substrate, we can actually use ethylamine that was created previously by alanine decarboxylase. So it turns out that glutaminase, the same enzyme that's responsible for catabolizing glutamine into glutamate, it can also react with glutamine and ethylamine instead of glutamine and water. And in the process, the ethylamine will attack this carbonyl carbon right here and displace the ammonia, creating L-theanine. So that's a one-step reaction, but it's still going to require ethylamine from alanine decarboxylase. So in a sense, it's the same number of enzymes required. The benefit of doing this reaction is that it does not require ATP. Okay, the prior one does. All right, now to contrast theanine to how GABA works, we need to have a brief understanding of what GABA is doing. All right, so this up here, this in green, this is a GABAergic neuron. That is a neuron that synthesizes GABA and releases it into a synapse. The synapse with its postsynaptic neuron. All right, so basically what we're going to look at is we start with some glutamate right here. So GABAergic neurons have to have glutamate because GABA is made from glutamate. So that's done through the action of glutamate decarboxylase. So glutamate decarboxylase removes a carboxyl from glutamate and makes GABA. As we know, the GABA is packaged into vesicles and it's exocytosed into the synapse, where GABA can exert its effects on a postsynaptic neuron. Now, the way GABA is rid from the synapse is it diffuses away to a nearby glial cell, and it's uptaken into that glial cell. We talked about GABA metabolism in a previous video, so we'll just briefly run through this, but suffice to say, GABA is shunted into the Krebs cycle through two enzymes. Those are going to be GABA transaminase and then succinate semialdehyde dehydrogenase. And that's going to give you succinate, shown here as succinic acid, which is a Krebs cycle intermediate. Through continuing through the Krebs cycle, succinate can be converted to alpha-ketoglutarate, and then through a transamination, alpha-ketoglutarate is converted back to glutamate. The glutamate can then be converted to glutamine by glutamine synthetase, and it's that glutamine that is transported back into the GABAergic neuron, where glutaminase undoes that reaction and converts the glutamine back to glutamate, and we're back to square one. The GABA can be made from that glutamate through the mechanism that we had just shown. So you have these GABA receptors right here, and they normally bind GABA. And in another video, we also talked about how GABA is required for the action of the GABA receptor, obviously, but that things like barbiturates, benzodiazepines, and alcohol can actually enhance GABA's effect on its own receptor. So if you have GABA, there's a certain effect you have, but if you have GABA plus ethanol or GABA plus a benzodiazepine, the effect of GABA binding is amplified because GABA binds tighter to the receptor, okay? So if I were to do something that said antagonize this receptor, that is going to promote drowsiness. It's going to promote sleepiness. Sure, you'll be more relaxed, I suppose, but you'll have the effect of sedation. So how does theanine differ from just a simple GABA agonist? Well, theanine doesn't do anything with a GABA receptor. Theanine has two modes of action that we're going to talk about. But overall, theanine is going to cause GABA to be predominant over glutamate in the central nervous system, which is going to promote the relaxation. Sometimes it's theorized that glutamate excess in the brain causes anxiety and things like that. And that actually makes sense to some extent. I mean, that would have to be studied further, but it certainly makes sense. So if you increase the ratio in the direction of GABA, you may promote more relaxation. But what theanine does is, number one, right here. Over here we have a glial cell. It's a very similar makeup over here. Glutamate in the glial cell is converted to glutamine by glutamine synthetase. The glutamine normally will be transported from the glial cell to the presynaptic cell. Okay? And this is not a GABAergic neuron. This is a glutamatergic neuron. This is a neuron that releases glutamate. However, to get the glutamine into the presynaptic neuron, you have to have a glutamine transporter. It turns out that theanine resembles glutamine, but it blocks that transport. So the glutamine cannot get into the presynaptic neuron. Well, what's the consequence of that? Well, in order for this neuron to have more glutamate, it has to get glutamine first. And the glutamine is then deaminated by glutaminase to make glutamate. So if you can't get glutamine into the cell, you also don't get much glutamate, okay? And therefore less glutamate is released by that neuron. So that is one way that theanine decreases the amount of glutamate available in the central nervous system. The other thing also is that what glutamate does normally is it has a receptor called the NMDA receptor. That's the main one. The NMDA receptor is the main glutamate receptor in the central nervous system. It's the most common one, and when glutamate binds to an NMDA receptor, there is subsequent depolarization of the membrane, and that just leads to activation of that neuron. Well, if you have lots of anxiety and you're very restless, etc., a theory as to why that is because you have excess depolarization of neurons in the brain, too much neuronal activation in the brain. Well, if glutamate normally binds to that receptor and promotes that activation, then theanine antagonizes the NMDA receptor, meaning it blocks glutamate from binding to the receptor, but if it blocks it, then glutamate can't bind. So theanine is more of it's like a receptor blocker. That's exactly what it is. It's a receptor blocker that prevents activation of these neurons right here, the postsynaptic neuron. And therefore, it doesn't act to inhibit the neuron, it prevents its activation. So theanine's modes of action are more indirect. It prevents glutamine from getting into the presynaptic neuron, so you can't get glutamate in any case, and then the glutamate that is there can't exert its effect because the theanine is blocking the NMDA receptor. All right. So those are the modes of action of theanine. Now, what we also want to talk about is, I mentioned at the very beginning of this presentation that GABA really was not shown to be very effective. And why is that? Well, it turns out that this structure of GABA, you can't really see it because I got it off of Wikipedia. But what you have to remember is at physiological pH, this amine on the left side is protonated, so it has a plus charge, and this carboxyl on the right side is deprotonated, so it has a negative charge. So overall, GABA is charged. It's charged. Now, this is the synthesis of theanine without the enzyme shown, but this molecule over here is theanine. Now, what you have to remember is, yes, it has an amine and a carboxyl group, but they're very close to each other, so they form a salt bridge. Whenever you have the salt bridge on an amino acid, that has a tendency of decreasing the polarity some because they're close together and the charges essentially attract each other, and for reasons that we're not going to go into, that decreases the polarity of the molecule versus having the amine and the carboxyl farther apart. Okay? Also, this functional group right here, the amide, doesn't have any net charges on it. Okay? And then it ends in an ethyl group, which increases the hydrophobicity. Overall, what that does is it means theanine is more hydrophobic than GABA. GABA is much more polar because it doesn't have any way really to cancel the charges, and it's overall more charged to begin with. It has a higher charge per unit volume. Theanine is more hydrophobic. It's not absolutely hydrophobic, but it's certainly more hydrophobic than GABA. So what they've shown actually is that GABA actually, if you would actually take it as a supplement, you actually can't get it into the brain neurons. And why is that? Well, but we have what's called the blood-brain barrier. The blood-brain barrier really, for the most part, allows more hydrophobic things through than polar. Since GABA is polar, if you intake GABA through the diet, you can absorb it certainly, but it won't go into the brain because it can't cross the blood-brain barrier. That's why the GABAergic neuron has to synthesize the GABA from glutamate, because GABA is not a dietary thing. However, because theanine is more hydrophobic, it has been shown that theanine crosses the blood-brain barrier. All right? And I will say this, I take theanine on an everyday basis, and I can tell you that it actually does work. It's not sedative, but it does tend to reduce anxiety quite a bit, so I'd recommend trying it. This is obviously not a sales pitch, this is educational, but I can just tell you from experience it works, and this is why. Theanine can cross the blood-brain barrier, while GABA cannot. All right? So hopefully that makes a little bit of sense. I just did this video because it's rather interesting. Hope you learned something. Make sure to like this video and subscribe to the channel for future videos and notifications. Thank you very much.